Morning, guys. Good morning, Akshaya, Vijay Mohan, Arshia, Sridaksha, Durga, Shanmati, Anuvarshini, Rupa, Vishalini. Good morning. Welcome to the other class. Good morning. Good morning. So what about others? Still they have to join, right? So you guys had breakfast? Or still on bed like this? Online classes advantages, guys. Morning. Okay. Shamli, good morning. Rupa. Oh, you didn't have Rupa? Okay. Have it by 8 because you guys have continuous classes till 12, right? So we will get break, but you guys will not get break. 10 minutes time may not be sufficient. So that's why we gave even 10 minutes gap with every, what is this? Every class. And even you can eat and see also. It's not like class, right? So you can't claim that you didn't have Rupa. You can have it right now. Just what I'm doing daily is that the same thing, right? I'm having along with you guys. Okay. Ashraf, good morning. Okay, guys, only 11 members came. What about others? So we'll wait one more minute and we'll start. Is my voice? I think my voice might be audible, but I see that there is no network issue, right? It's like the screen is not freezing, right? For me, it's like it's a little bit freezing. I don't know what's wrong with this. Okay, guys, we'll start. So we had enough wait. 5 minutes we already wait. So let's start with begin with the class. So today we are going to discuss two lessons. Is strategies for enhancement of food, right? And then one more is biotechnology principles and processes. So I gave equal importance for both the lessons because if you see that in these two lessons you will find a lot of concepts will be there. Okay. So shall we start now then? Everything is clear, right? Okay, great. So I've given 2 days for these two lessons. So Thursday you have only two lessons to write test. Especially these two lessons are very much important, very important as well. When it comes to biotechnology processes, what happens is this. In this, you will be getting important concepts, about the vector, plasmid, right? The RDNA technology is very important. So that's why I kept 2 days. So let's dive into the topics and then we can go ahead with. So we'll start with biotechnology and biotechnology principles and processes. Okay, because strategies for enhancement of food materials. This the chapter you have enough revision, but we will touch the important aspects also. Before that, let me check the other lesson, that is biotechnology. So how far is your understanding, so that we can go ahead with, because I know your understanding on the strategies for enhancement, but I don't have any clear cut idea about what is your take, or what is your understanding of this lesson, this biotechnology lesson. So let's start with the biotechnology. There is principles and processes, right? So when we talk about this lesson, right? So we will touch up the important aspects in this lesson. So begin with the what do you say, the definition of biotechnology. It's very easy that when we use technique, right? Where we use either living organisms or their enzymes, right, to produce some products which are useful for mankind, right? So, for example, you take microorganisms, right? So, microorganisms, or you can take their products, enzymes. So, those products and processes which are useful for the mankind is what we call it as biotechnology, where we use integrational approach in this biotechnology. For example, if you want to, if you want to produce a large-scale vaccine, for example, peptide vaccines, right? So how do we do that? So for that matter, we will see the applications in the next lesson. But now we will see how can we really produce our desired gene products, right? That's the important aspect of this lesson. So let's move into the principles one by one. So, what are the core techniques that are involved in this modern biotechnology? Right? When it comes to core techniques, we use genetic engineering as a core technique in this biotechnology. It means the genetic material DNA or RNA, DNA and RNA is chemically altered, right? So there is an alteration process that is occurring, right? And then introduced into host organisms to change the phenotype. So, in principles of inheritance and variation, we already talked about genes, characters, traits, genotype, phenotype, right? So, the enzyme that is produced and the processes or the other products that are produced by microorganisms is something we call it as phenotype, right? So that phenotype can be altered at the gene level, right? So that alteration of gene, right, the genetic material is nothing but one of the process we get is genetic engineering, right? So this is a two core techniques that are involved in this modern biotechnology. One is genetic engineering and the other one is maintenance of sterile ambience. It means like how do we really process later, right? So once we get that chemical out, so that chemical, uh, chemical engineering processes. For example, to get the desired, products like antibiotics, vaccines, enzymes. So there is a gene alteration at one end, right? At the same time, once a product is forming, we will use chemical engineering processes for growing desired microbes or eukaryotic cells for manufacturing of antibiotics, vaccine, enzymes, etc. It means these we touched these aspects in microbes in human welfare, right? So those are all the products are produced by this core technique, one of the biotechnology technique. Okay, so these are the two basic core techniques that are used in modern biotechnology. Is it making sense? Is it making sense, guys? Is it making sense? Voice Ashraf, Janil, Mrutula, many members, they didn't come. Guys, we'll see one more week. Okay, we'll see this week whoever not attending the classes and whoever not giving the test, we will be simply removing from the group. No more discussion. That's it. I will talk with sir also, because we are putting such a lot of efforts to reach you guys in the home setting, and if you guys are not able to come, right, you guys are gave names and numbers as you guys are all interested. So please do tell to your friends also. This week is the last final week. If you guys are not active in the participating in the tests and classes, we will be removing. That's it. As simple as it. Okay, fine. So let's go into the next aspects, that is basic steps in genetically modifying an organism. For example, so here we have two core principles that we are talking about. One is core techniques. One is genetic engineering, that alteration of genetic material, DNA and RNA, and then maintenance of the second aspect is the chemical engineering methods, and then maintenance of this microorganism, bioreactors. These are all comes under second core techniques. And then next what we have to talk is basic steps in genetically modifying an organism. So how do we really modify an organism? Right, these are the basic steps we need to follow. So one of the basic step is identification of DNA with desired genes. So here we will be talking about the genes, because these genes are altering the phenotype. So if we want to change the phenotype, we need to actually change the genotype. So what we have to do is, first we need to identify the gene sequence, DNA sequence, which is affecting this phenotype, right? So we have to first identify a desirable DNA, right? Or the identification of DNA with the desirable genes. So that sequence needs to be there, right? So in this identification of DNA with desirable genes, we have it's a kind of a we follow some traditional hybridization techniques, right? So which can actually explore. We can explore in this 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 traditional hybridization, using traditional hybridization techniques, right? We can actually manipulate the undesirable genes along with the desirable genes. So we know some set of genes are undesirable and some set of genes are desirable. So we can eliminate these undesirable genes and we can select these desirable genes, and then we can go ahead so that we can isolate them and we can introduce into new organism. That's what biotechnology. So genotype alteration is taken care by the genetic engineering, and this gene, once we identify the gene sequence in the DNA which are desired, and we know now that desired genes as well as undesired genes. So undesired genes will not select. So desired genes we can actually select, isolate, and then introduce into, right, target organism. So that target organism varies from the product, or what product we are actually looking for. The next step involved in this genetically modifying an organism is introduction of identified DNA into host. So host is what which takes up this DNA. The desirable DNA is taken up by the host. So a vector DNA such as plasmid is used to deliver an alien piece of DNA. It means, what exactly it means? What is a vector DNA actually? What are the vector? What are the vectors we use in this biotechnology? Vector. What are vectors? The one which has the direction and magnitude, or something else? Okay. Right. 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 Vector and host. Right. There shouldn't be any confusion. So I'm asking about vector DNA. vector DNA, Arshia, Sri Durga. Yeah, last. Yeah, correct, correct. Yes, guys. Yeah. Right. The organism which helps in transfer of DNA to host. Yeah, correct. Yeah. Actually, that's not an organism. That's a piece of DNA molecule which. Sri Daksha, we are talking about vector. Okay, so we're not talking about host here. Yeah. So let's move into the next one. So first, what we are doing is I asked a simple question, that is, what is vector DNA? Okay. For example, it is a plasmid which we use to deliver a alien piece. Alien piece is nothing but alien piece or desired gene or target gene. These are all different synonyms for the desired gene, which we use in our DNA technology. So second one is, first one is identification. Next one is introduction of this identified DNA into host. So how do we deliver into the host? By using a vector, right? Like plasmids, right? And then third one is maintenance of introduced DNA in the host and transfer of DNA into its progeny. So what we have to see is we have to maintain this, the DNA which is already, transferred into the host, right? And then we have to allow it to multiply so that we can get n number of copies, right? So these are the steps involved in genetically modifying an organism. So far, we are not discussing the steps involved in RDNA technology, right? So we are talking about, right, how do we modify an organism? How do we modify? Actually, this is how the DNA desired gene sequence we need to identify, and then we have to transfer it into the host with the help of vectors, and then we have to go for its maintenance, right? So we have to select this target DNA has, uh, you know, what do you say? Uh, we have to allow this alien DNA to be integrated into host DNA, so that once the host divides, even this plasmid also get divides. So in that way, we will be maintaining, and then we will be altering this organisms. So this is how we can alter or genetically modify an organism. So let's talk about tools of recombinant DNA technology. So we will talk about tools, right? So in these tools, we talk about the tools of recombinant DNA technology. What are the different tools we use? We use molecular scissors. What do you mean by molecular scissors, by the way? Molecular scissors. Yeah. Rupa, it's not just enzymes. Not all enzymes are restriction endonuclease. Yeah. Yes. Correct. Okay. All of you correct? Yeah. Rupa, you need to. Yeah. Rupa, correct. Akshaya, Arshya, Sri Durga, Vishalini, Nivedita, Daksha, Shanmati, Vijay Mohan, Sri Durga. Yeah, all of you guys are correct. Okay, let's move on. So we use different what do you say, tools in to attain this rDNA technology, or to actually within this rDNA technology or recombinant DNA technology. So these are the tools that we use, right? So what are the tools? For example, first one is we'll talk about restriction enzymes. Those are molecular scissors, right? And then we'll be talking about cloning vectors, and then we will be talking about competent host. So three aspects we need to have, three tools we need to have. One is restriction enzymes we need to have to cut the desirable gene, and then we will be talking about the second aspect. The second tool we'll be talking about is cloning vectors. So once the desired gene, so if you think, if you see this cap as a desired gene, right? So this is a plasmid. Okay, so this desired gene, once it is cut from original sequence, and this is the gene we need to produce, or the gene products we need, right? So this gene needs to be identified and then isolated, and then it needs to be transferred, right? And then it needs to be inserted into the vector, and then this vector would actually transfer into host. So next step we'll talk about the cloning vector with the desirable genes. Once this cloning vector is ready, right, artificially we can create a vectors as well, right? We'll be talking in this lesson, NCERT text, they gave it as PBR 322. So like that we have several vectors. So this vector needs to go into the cells, right? It means host cells to produce the products. So that is a third step, that is competent host. So here we use the word competent, right? So that is a important aspect for whether this plasmid actually transformed this cell or not, right? Host. Based upon that, we will say they are competent cells. They are ready to take up this plasmid with the desired gene. So that is a third tool we will be using. So in this lesson, your focus needs to be on correct, incorrect statements of the processes. For example, 4 techniques. So I'll be giving yesterday I gave I by mistake I sent daily practice file twice in the group, instead of sending outline of the chapter human reproduction. Okay, I will send the link here. You can download all the materials regarding our classes. Okay, and sorry for that. And then today I will send this principles of biotechnology, so that you will get to know. So in these principles, you may get correct or incorrect statements. So you should be extremely careful while you are reading. Okay. And then you may get the jumbling sequences also. So they may give 1, 2, 3, 4, 5. Actually a process needs to be a sequential order. So they shuffle that order, and then they ask you to arrange. So these are the questions you may get expect from the principles. Okay, not just from whole lesson. I'm talking about principles, right? The basic principles, and then 4 techniques. Then when it comes to tools of recombinant DNA, the first tool is restriction enzymes. Second tool is cloning vectors. Third tool is competent host. So first we will talk about restriction enzymes, molecular scissors, and what are the functions of molecular scissors, and what are the types of molecular scissors, and what are their restriction sites. Why they are called molecular scissors? They don't just randomly cut at any sequence. They have a specific restriction sites for each of the restriction enzyme, so that we can use the same restriction enzymes to cut the desired gene as well as to cut the vector, so that we will get a similar opening. Are you able to understand? Right? So this is a vector and this is a desired gene. So this desired gene will cut at specific sequence using, for example, EcoR1. So if you use that EcoR1, right? EcoR1, when we use this EcoR1, it cuts at specific sites, and the same EcoR1 can be used in cutting the vector as well, so that this can perfectly fit into the desired gene, can perfectly fit into the vector. That's why in this restriction enzymes, especially the tools of recombinant DNA, first two restriction enzymes, you need to focus on the names of the restriction enzymes and their sites of where exactly they cut. Okay, so those are the important aspects. Okay, especially even you may be getting confused with what are exonucleases, endonucleases, right? So exo means something outside, right? Exoplanet, exo, right? Endo means inside. So if you see this exonuclease is nothing but they remove nucleotides from ends of the DNA. So if this is an piece of DNA, right? So for example, this is a piece of DNA, my hand. So this is entirely a piece of DNA, and this is unwanted. We don't need this, right? So this exonuclease, the restriction enzymes which has an exonuclease function, will remove these nucleotides. DNA is nothing but it's a sequence of nucleotides. From ends of the DNA, this piece will be cut. So when it comes to endonucleases, these endonucleases would specifically remove, for example, if I have to remove this watch, it means this is a DNA sequence, right? This watch is also within the sequence. So I want specifically remove this watch. So I need a specialized enzyme to really remove this watch. So we use restriction sites. That's why we call endonucleases. So endonucleases, they cut at specific position within the DNA. So this is, if you consider this is a DNA and this is a sequence we need to cut, right? So this sequence will be removed within the DNA by using endonucleases, right? So they bind to specific restriction or recognition sequences of DNA and cut the two strands at specific points. So you have DNA means two strands, right? Two strands, right? So they cut at specific sites. So that's how exonucleases, endonucleases. Likewise, you have EcoR1 and HIND2. HIND2, what is the function of HIND2? Where exactly it cuts? How many nucleotides or how many base pairs it cuts? For example, in HIND2 is 6 base pairs. 6 base pairs it can cut, right? So, for example, the first restriction endonuclease, that is HIND2, it cuts DNA molecules by recognizing a specific sequence of 6 base pairs. So within a this 6 base pairs are very specific to HIND2. So wherever the 6 sorry, so wherever the 6 base pairs are there, they will be cut by this HIND2. As simple as that. Okay. And then coming to the next aspect of this restriction endonuclease functions, right? Direction also important. For example, it is a sequence of base pairs that read the same on two strands. For example, if you take palindromic sequences, right? Palindromic nucleotide sequences are present in the DNA, in which they has base pairs which we can read same on two strands. It means 5 prime to 3 prime, this is a direction, 5 prime to 3 prime, and 5 prime to 3 prime. If it is A, T, G, C, exactly you can read from this direction. For example, GAA, TTC. Okay. So 5 prime to 3 prime, if you have GAA, TTC, so at 5 prime to 3 prime, the alternate sequence will be a palindromic. Are you able to understand? Are you able to follow, guys? I'm talking about palindromic sequence. GAA, TTC, right? 3 prime. So 3 prime is CTTAAG. So this is what I'm talking about, palindromic sequences. So 5 prime to 3 prime, 5 prime to 3 prime, you will get that. So these palindromic sequences are very much important when it comes to, and this in this recognition enzymes, this restriction endonucleases would recognize these palindromic sequences. That's how we wonder at this molecular level, this much of complexity there with this endonucleases, and then that's what we have to. It's an hats off for the scientists who actually gave all these techniques as well, right? Very precise actually. So these are the one of the what do you say restriction enzymes functions, recognition of palindromic sequences, and then these restriction endonucleases would actually produce a sticky ends. It means if this is an DNA molecule, right? When the restriction enzyme acts on this by recognizing palindromic sequences, or if it is a band 2 or hinge 2, it recognizes specific sequences. And especially when we talk about ECOR1, let us take ECOR1. So when this enzyme we add into the to break the to open up the vector, it specifically cuts at specific sequence and produces sticky end like this. So here there is a gap over here, there is a gap over here, so that what happens is the vector DNA can actually the vector DNA will produce this sticky ends, so that the desired gene can actually go and integrate within the vector. So that's the one of the important aspect of the restriction enzymes, that is they produce sticky ends. Right, there was a beautiful diagrams given in NCERT as well. So we'll have foreign DNA, we use restriction endonucleases. We have a vector DNA plasmid, we use the same restriction enzymes, and we cut at specific ends, and then we ligase, and then we will allow it to, or we will deliver into the host, and then that process we call it as transformation, right? And then so before transforming, we need to identify this DNA fragment. So for that DNA fragments identification, what we will do is we will separate and isolate DNA fragments, right, by the one of the technique called gel electrophoresis. So they may give this diagram that is given in your NCERT as well. Okay. So you need to remember even the stain that we use, ethidium bromide. Okay. So when this, the stain we use is ethidium bromide. At the same time, we will expose these bands to the UV radiation. So in this way, what we can do is we can actually figure it out. We can actually see this separation of DNA fragments, and then we can select by process of elution. Okay. So elution is one of the important aspects that you need to cover within the restriction endonucleases, identification, isolation of DNA fragments by the process of, by technique that we use is gel electrophoresis, and the dye we use in gel electrophoresis is ethidium bromide. Okay. So this is one of the aspects that one of the tool that we are discussed, that is what is it, restriction and enzymes. And then the most interesting part would be the competent cells, that is blue white screening, right? The blue white screening, how do we actually produce competent cells? So this production of competent cells that usually we come across in MSc syllabus, and we use it in our laboratory during our research. So we produce these competent cells, so that they will be readily taking up this vector, the plasmids, with the desired gene. So that is the most interesting part within this tools of recombinant DNA. And talking about the cloning vectors, the cloning sites, and where we need to introduce our DNA, desired DNA or alien DNA or target DNA, right, is the more tricky part, right? So based upon that, we will actually even design the plasmid. Okay. So that's one of the aspects, and then let's move further, right? Within the cloning vectors, right? What are the we use here plasmids, right? So if you consider this plasmids, right? So the second tool we are talking about is plasmids. Okay. So, sorry, the second tool we are talking about is cloning vectors. So, within which we will have a different vectors. So, when we talk about vectors, right, what are they actually? So, it is a DNA molecule that can carry foreign DNA or alien DNA or desired DNA, right? The segments, right, and eventually it can replicate in the host cell. So if you consider this is a vector, right, and this is a desired gene, so in this way, this is what we want to produce, a vector with a desired gene. So when we introduce this into, when we introduce this vector into, what do you say, a competent cell, assume that this is a competent cell. When we introduce into this competent cell, they reproduce and then they divide into two, right? So likewise, even the desired gene products also will keep on increasing, right? So within this cloning vectors, right, what we can get examples, someone already gave bacteriophage an example. Yeah, that is correct. And we use plasmids as well actually. So plasmids are autonomously replicating circular extra chromosomal DNA. For example, if you see a bacteria, right? So there is a genetic material for the bacteria. At the same time, there is an extra chromosomal, not belongs to chromosome. It can autonomously by itself it can replicate, right? So that DNA sequence, what we call it as plasmids, and they may vary the copy number. Maybe one cell may have one or two plasmids, or others may have maybe around 50 to 100 copies as well. Okay. And coming to bacteriophages, so uh, these bacteriophages they will be having very high copy numbers, right? Because they are not cells, right? So they have this genetic material, right? And within this bacterial cells, this bacteriophage can actually produce a larger, more copy number, because during molecular base of inheritance, we already discussed about to prove how we prove that DNA is genetic material. We use Hershey-Chase method, right? So bacteriophage and bacterial cell we took. So in that sense here, so this bacteriophage can actually eventually replicate more in number, and so they will have more copy number. Okay, so that is one of the aspect. Yeah. Okay, let's move further. So next we are talking about the features of, that are essential for, hello. Yeah. Okay. Yeah. So what are the features are required? Okay, the features required for cloning into a vector. So this, how do we actually we can know what are the different features that are involved to produce a vector? So what are the aspects that needs to what are the features does this vector needs to contain? Because the whole genetic rDNA tech is based, completely based upon these vectors, right? So this vector needs to be having certain features. What are those? So when we talk about vectors, especially they have to contain certain features, like one is origin of replication, ORI. So they need to have origin of replication, and then they needs to have a selectable marker, right? Selectable marker in the sense, like based upon these markers, we can tell whether that cell has transformed or not transformed, right? Transformed or non-transformants can be distinguished based upon the selectable markers, right? So these selectable markers for organisms, for example, E. coli includes, ampicillin, right? Chloramphenicol, tetracycline, kanamycin, right? So these are all the selectable markers. It means, so this is a tricky part actually. Why? Because we introduce our target or specific DNA into these regions, so that what happens, if this is a DNA, right? For example, this is with this I will stop here and then I'll start the other lesson, because this is a very important aspect. So I wanted to make sure that we will start this again tomorrow, so that it will be very clear, so that you can study till still till half of the lesson you can go through, and tomorrow we can continue. So most important aspect here that everyone may confused is transformant and non-transformants, right? Can you guys tell what is a transformant cell and non-transformants? What are transformants and non-transformants? Which cells are called? Transformants transfer an alien DNA into host. Okay, so what defines and transformant? Why we call transformant cell as a transformant? Transformants. Host will accept the foreign DNA. Right, correct, correct, correct. Where do we introduce our target genes? Yeah, cloning sites. Okay, fine. Cloning sites. Yeah, recognition sites, cloning sites. Okay. Yeah. So if you see this, for example, uh, yeah, yeah. Anyone else? Okay, right. So tomorrow we'll talk about these aspects, cloning sites, and how do we produce competent cells, as well as what are transformants, non-transformants, what are transformants with the recombinant plasmids, transformants with no recombinant plasmids, and non-transformants. All these aspects will cover. At the same time, how do we screen them? How do we really isolate? How can we tell that these cells are transformed or not transformed? On what basis, right? How do we see the colony structure, right? How do they appear? Blue white screening, we say, right? All these things we will talk tomorrow. So with this we will stop this chapter. That is you can read this outline of the chapter till 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 the tools of recombinant DNA, that is restriction enzymes. And the second one, that is cloning vectors. From cloning vectors, and the remaining lesson can be completed tomorrow. I will take tomorrow. So let's start with the next chapter, that is strategies for enhancement of food. Okay. So is that clear? Okay, guys. Okay. Prop 9. Right. The half lesson today, today's half lesson, and the biotechnology as well as the strategies tomorrow also will cover. So likewise we can finish it off. Okay. Because two lessons you need to go through, botany and zoology. Otherwise I would have took only one lesson completely finish and then tomorrow one. But we are learning as a botany and zoology, right? So we have to go through, parallel. So when we talk about this, the next lesson, strategies for enhancement of food materials, food production, you need to have more focus on, when comes to this management of farms and farm animals, right? Dairy farm management, beekeeping, as correct or incorrect statements. Okay, so you need to be very much careful about these statements that are given directly in NCERT. Okay, no need to refer any other book. First you go through the NCERT as well. First NCERT and then any other book. Okay, so when we talk about this management, right? This management, or dairy management, poultry management, or uh, uh, in this you have to focus on the what are the statements, true or false, in the sense correct or incorrect statements you need to be covering. So within the animal husbandry, so these are the only aspects that they may ask from this topic. Okay, and then what do you say, the numbers like world livestock population, right? So that is 70%, right? Farm produce is only 25%. So these numbers are important, right? These two numbers are important. So when it comes to management of farms and farm animals, they can be asked as correct or incorrect statements that you need to once go through. Okay, it's like self-explanatory. So I no need to waste time right now. Okay. So when we talk about the next aspect is the animal breeding is important aspect in this lesson, and the definitions of each one: outcrossing, outbreeding, inbreeding depression, interspecific hybridization, and its examples. So these are the aspects you need to cover more, right? Especially you should not get confused with the definitions like breeds, what is a breed, etc. So in academics itself we have an enough revision on this and explanation. If you have any further doubts also, you can reach me any point of time through WhatsApp, okay, or any means, call or whatever it is. Right. So in animal breeding, we talk about the breed. Breed is nothing but a group of animals related by its descent. For example, parents are there, and then there's a family tree, descents, and similar general appearance, features, and size. So this is what we call it as breed. So when we talk about breeding, we have different techniques we use in breeding, right? Where why we are actually breeding, it's a sort of artificial selection we use here. At the same time, we are humans, we creating different breeds so that we can enhance the food production, because to meet the growing demands of the food and the nutrition and other aspects, we need to make sure that humans do have a adequate food supply. So that what we are doing is that we are even producing the organisms with the very uh, what is a conventional techniques to what is a modern techniques we use in this breeding. So breeding techniques vary from conventional to modern. So let's see what are the different breeding techniques we use. For example, if we take inbreeding, right? So inbreeding is nothing but it is a mating of more closely related individuals within the same breed for over 4 to 6 generations. So for 4 to 6 generations, they will be keep inbreeding among themselves. So what happens is, so you will be able to produce some. For example, this breeding would takes place with the help of desired characteristics, like for example, high milk yielding and the immunity, right? So these both desired characteristics would be coming in the offspring, and that would be kept the same homozygosity would be maintained by this inbreeding technique. Almost for 4 to 6 generations we use this inbreeding. So this inbreeding has both the advantages as well as disadvantages, right? The disadvantage of this inbreeding is that it gets the inbreeding depression. So inbreeding depression is very very important when and how do we overcome the inbreeding depression is what essential aspect in this lesson that you need to touch upon. So how do we come out of the inbreeding depression? So yeah, can anyone tell how do we come out of this inbreeding depression? So inbreeding is a technique where we use create a homozygosity over by mating within the same closely related species for over 4 to 6 generations, right? So how do and then it leads to homozygosity. At one point, it's a pro that it creates a similar kind of an organisms with desired characteristics, but there are cons, disadvantages like inbreeding depression. So how do we really answer this inbreeding depression? So how do we come out of this inbreeding depression? By single outcrossing. Okay, so what we do is simply to solve this problem, we'll be selecting animals. This animal should be mated with unrelated superior animals. So not within the family tree. So we have to go unrelated, not closely related, unrelated, but within the same breed. So that you need to remember. So if you take this is family 1 and family 2, right? Family 1, family 2, but these both are coming under. So if you take this like this, right? For example, if you take these are the two families, right? So these two families belongs to my this breed, a single breed, but here the inbreeding depression is created in one family tree and here not. So we have to mate these organisms, the homozygosity which we created, and this should be also superior animals, but they are not closely related, but they are distinctly related, but within the same breed. That's how we come out of the inbreeding depression. Yeah, correct. Excellent. Correct. Correct, guys. Yeah. And then we'll be how do we do this outbreeding? So it is a breeding of unrelated animals. So from now what we see is the breeding choices that we make would be differing from within the family tree to the different species. Okay, so that's how we put this gradation: inbreeding, outbreeding, and then interspecific, intraspecific hybridization. So outbreeding is something a breeding of unrelated animals. It includes two processes: outcrossing, crossbreeding, and even interspecific hybridization. Okay, three different aspects includes in outbreeding, right? So it includes outcrossing, crossbreeding, and then interspecific hybridization. So these are the if you see these outbreeding techniques, the organisms which came from this crossbreeding examples are important. Interspecific hybridization. right? For example, it is a mating of male and female of different species, right? Intra means within, interspecific, so two different species and they are mated together. For example, mule, right? Right. So the mule, which is an offspring, right? So that is produced by interspecific hybridization. Two different species mated to allowed to mate together, and then they produce this, hybrid. So right from very closely related to different species itself is what breeding techniques would we can choose on. And here, in this aspects, we need to touch upon the examples. What are the interspecific hybridization? Like animals which are produced through interspecific hybridization. What are the organisms which are produced using crossbreeding? What are the which organism we use is inbreeding, right? So these are the aspects you need to focus on. Is that clear? Ma, are you guys following, Ma? Is that clear? Yeah, yeah, yeah. I told right yesterday I forgot to send one of the outline of the chapter, the human reproduction, where you can where you can get in this link. Okay, so use this link. Uh, what I will do is I will create a feedback form. Okay, I will send it at the end of the week. Okay, I need the feedback for the classes as well. Okay, how do you really find this is useful? Because this is not an ordinary setup of the class. It is very unusual, like unusual in the sense like we didn't really expect that we have to take classes in this way. If it's a classroom teaching, it would have been a different story, right? The plan would have been very much different. But this is what we can really do right now. But I'm doing or we are doing 200% what we can do. But you are at the receiving end. You guys are all at the receiving end. You need to receive as much as possible, and you have to attend the tests and classes for sure, so that we can track our progress and track your progress as well, so that you will be more confident. Okay. So with this, I will please download download that material within that link that was given. Today's class outline and the 3 different worksheets will be provided shortly. And thank you guys for your attention. I'm really happy to interact with you guys. Hope you guys are really enjoying these classes, and it's really you if you I think you guys are finding it very useful as well. So please make sure that you ask doubts at any point of time without any hesitations, or don't hesitate to ask any sort of questions. Okay, please reach me. Thank you guys. See you in the next class. Thank you.